Yeah, that you can go to the next slide. Okay, so yeah, I'm going to talk that. There you go. Okay, so what I'm going to talk about what are bad sequences, and the work that we've done on FunGCat has informs this, basically this entire presentation. So one of the conclusions was that encoded biological threats are primarily, and also this is just in terms of sheer numbers, primarily from microbes that cause infectious diseases, and secondarily from metazoan, that's animal mostly, and plant venom components and toxins. So on the pretty pictures on the screen, it differentiates some kinds of toxins. So ciguatoxin, you see down there, it's like the figure, the complicated chemical figure. That's not an encoded toxin. The organism, I think it's adenoflagellate, needs to build that thing from different precursors. On the other hand, dendrotoxin, which is from the handsome green mamba above the ciguatoxin figure, that is an encoded sequence, and dendrotoxin is a really nasty toxin. The middle picture shows a castor plant, including the seed pods, and of course ricin comes from castor plants, and those pretty seed pods. And if you mesh them up and everything, you can make ricin, and that is a sequence. While the trachotoxin and tetrodotoxin, those aren't encoded sequences. Next slide, please. So, can we go to there we go. Okay. So bad bugs essentially are parasites. And of course you have to differentiate what are they, the parasites infecting what. We're mainly concerned about, and as Matt referred to this, we're mainly concerned about those that affect the stuff that humans are interested in. So us and our livestock and our crops are the main things. So there are different kinds of parasites. You can have the ones that fall into the microbial category are viruses, bacteria, a lot of fungi. I don't think that one in particular, and protozoal parasites. There's also worms, like multicellular parasites, but I'm going to talk a little bit about worms. Actually, I'm not going to talk much about worms at all, except maybe on the next slide. So, and these are things that live in or on a larger host and cause disease, which is loss of homeostasis to the host. Yeah. So how many bad bugs are there? So there's 1 trillion microbial species on Earth. That's microbes. That doesn't include the worms. Of them, 1,300 roughly, and this is my own accounting from the literature. There's 1,300 viruses, bacteria, fungi, protozoa, and worms that can infect immune normal, that's a term of art, immune normal humans. 300 of those are worms, so 1,000 of them are microbes. An additional 700 bacteria and fungi can cause disease in immune compromised humans. That's about 4% of the population. Immune compromised includes folks that have some barrier dysfunction or have HIV or are taking immune suppressive drugs for transplants and things like that. Or there's other diseases that can lead to immune compromise. Now, if you're of that tiny percentage of people that has severe combined immunodeficiency, which is essentially no adaptive immune system at all, and because of the defects, the genetic defects, hardly any innate immune system, then there's probably billions of microbes that can cause disease in that sort of human. So the susceptibility of the host in terms of how good its immunity is does determine like kind of how bad the bug is, or like what things can harm us. Next slide, please. Okay, so how to host resist parasite. Obviously, I've given something a little bit. So we've got barriers. We've got physical barriers that protect us, the skin, and the gut epithelial from things that are going to try to get in. There's lots of ways that microbes have to subvert those things and to get around them. We have chemical barriers: stomach acid, mucus, antimicrobial peptides that are secreted by our immune cells. And then there's biological barriers, in which case like the microbiome in the skin and the gut. And other places. Okay, so that's one component. But the biggest component, well, I can't say the biggest component, but a really important component is the immune system. And there's two kind of flavors of the immune system. There's the innate immune system, which is found in literally all organisms on Earth, except for viruses. And it's constant, it's invariant, And then vertebrates, on top of vertebrates, have an adaptive immune system, which has one of its housing is in our long bones, the arm, the leg, and that develops over time, and it's specific to the malady that's afflicting us at the time. Okay, so and the figure shows basically a bunch of immune detectors and where they are on a cell. This is almost, I think, nearly every cell in your body is has these things on them. Some of them are specific for bacteria, bacterial proteins, others for fungal, and they target the innate immune system recognizes things that are common to all microbial life and also some worms. All right, so to be a parasite requires a susceptible host, but what makes a host susceptible? Yeah, okay, and the sequences concerned with immunity comprise about 6% of the human genome and are under the greatest selective pressure of all the genes that we have, which means that they are constantly changing. Why? Because this is a constant thread. Next slide, please. Yeah, so the outcome of the encounter between the host, us, and the parasite is chiefly governed by the immune defense of the host, all those molecular components and physical components that comprise the immune system, and the virulence factors of the parasites. What would happen if we lacked an immune system? That's kind of answered in the SCID example I gave above. And our hypothesis is that sequences that subvert innate immunity essentially they force the host to be susceptible to the parasite. Why is this important? Because we have to look at the function of what the sequence is doing. Next slide, please. Yeah, so I'm going to talk about immune suppressant sequences or immune subverting sequences. So there's a lot of sequences in microbes, thousands of sequences in microbes that suppress host cell immune signaling. So that if they can suppress the instigation of the immune response saying, hey, there's something here in me, maybe somebody should look at it, send it back and let the immune cells come in. If they can suppress that, then they've gone a long way toward causing an infection. Another thing you can do is subvert antigen presentation. So once some of the bad bugs have been chewed up, pieces of them can be displayed on the outside of the cell, like I'm doing with my hands here, and they've got peptides in here, and then the T cells and other things can come and look at this and say, oh yeah, that's a parasite. We're gonna come and kill you now and stop the infection. Then there's other immune effectors that can be countered, which I'm going to talk about in the next slide, which shows, I think, streptococci. Okay, so this is some example of, and I'm not going to, I know a lot of you are bioinformatics people, this is just going to make you crazy, but this shows some of the effectors that our immune system has, and it shows some things that this streptococci, a particular kind, a genus of bacteria, can do to avoid them. So you can make these neutrophil extracellular traps in the left-hand corner, degradation of nets, and in response to being trapped in these nets, webs of DNA, is that the streptococci will secrete these proteins, specific proteins, to degrade that DNA and chew it up, and so the streptococci are liberated from their prison. I think I mentioned before that there's antimicrobial peptides that our immune cells can put out, and other cells, not just immune cells. And there are ways that the streptococcus says with SPB and SIC that will neutralize those, cut them in half, and do other things to those antimicrobial peptides so they can't reach the bacterial surface. Oh, sorry, go back. Or do you want me to go forward? Sorry. Yeah. Okay. So I won't. So there's ways that the bacteria have to frustrate complement, which is a way that the bacteria and fungi can be recognized and killed. The streptococci can destroy antibodies in different ways. They can also degrade chemokines, which are a way that the immune system signals, and they can target directly the neutrophils and other phagocytes that are trying to eat them up with nasty things like SLA and streptolysin O and streptolysin S. Okay, next slide, please. Okay, so I'm going to talk a little bit just how when we look for these things, when we built the database, how do we find these things? So we look them up in the literature, do search strings and stuff, and these are some questions we ask. Does it act on the parasite or the host? Is it extracellular, intracellular? How does it get to where it acts? Is it secreted? Is there some adherence and invasion? Is it like a toxin thing? How does it work? What is the targeted molecule that it's going after? And which host activity or pathway does it affect? And what's the general effect on the host? Is it messing up an organ? Is it destroying a tissue? Things like that. Next slide, please. Okay, so what are the functions? So adherence, if it doesn't adhere, nothing's going to happen if something doesn't adhere to the host. And then some proteins also enable invasion, to be basically being pulled into the cell that it's trying to attack. Next slide, please. I think that's dissemination. I could be wrong. Okay, no. So we're going to jump to damage, though. So the damage is a serious, if the sequence in question is causing damage, there's different kinds of damage that we specify. Cytotoxicity, it interferes with the cell's metabolism and kills it. It can disrupt the cell membrane. It can permeabilize tissue. It can destroy the extracellular matrix of the organism on which the cells are sitting. It can release the cell from the substrate, from the ECM. It just pops it off, and then the cell floats away. It can disable an organ, and it can induce inflammation. And that example is a T cell receptor, which basically turns on all your it makes a cytokine storm, and that's bad for you. All right, next slide, please. This is our little ontology, the little homebrew ontology for how we label these. So damaging SOCs are cytotoxic. They degrade tissues. They disable organs, or they induce inflammation. They can adhere, they can disseminate, they can enable host cell invasion, or they can movement in host cell. And so these are all ones that operate directly, and direct acting sequences target, hit, and adhere and do something directly to a host molecule. There's some that operate indirectly also. And immune subverting SOCs are also important direct acting sequences of concern, and they do a variety of things, as the streptococcal example I hope showed. Next slide, please. So there's 32 machine learning predicted functions of sequence of concern in SeqScreen. They're going to come out as an output. Krista's output showed, those, I think there were ones in those in that file she showed. And these are on GitLab on the on Todd's lab site. Yes. Yeah. And next slide, please. Yeah, so it appears as an output. If there's nothing there, if there's no funsoc for that, or if that sequence doesn't have a function of a sequence of concern for that entry, like disable organism, it shows up at a 0. But if it's got an entry like cytotoxicity for Bifidobacterium longum hemolysin, then it shows up as a 1 because it's cytolytic. Next slide. We've also got pathogenesis gene ontology terms. They were nice enough to let me contribute some sequences and some terms. It can be found at that website down there, and the slide is in the PDFs. Next slide, I think, gives some examples, or maybe not. I can't remember. Yeah, so here's some examples of in the last slide over there I can't remember if this is on the list that Chris showed. No, it's not. But anyway, but the PathGo terms give greater granularity of molecular function. So PathGo 278 is stimulate small GTPases in another organism. The 255 there is mediates guanine nucleotide exchange for a small GTPase in another organism, and the last one is modulates transcription in another organism. So that gives you a different level of, in addition to the effect that the funsoc show, it gives you some what's the protein being targeted in the host, which I think is useful. Okay, next slide. Okay, we're done.